Hello everyone, and welcome to Think Biochemistry. This channel is dedicated to making biochemistry simple, interesting, and easy to understand for students of pharmacy, medicine, life sciences, biotechnology, nursing, and other health science courses. Our goal is to explain complex biochemical concepts using clear diagrams, real-life examples, animations, and exam-oriented explanations, so that you not only score well in your examinations, but also build a strong conceptual foundation. If you're passionate about understanding life at the molecular level, you've come to the right place. So don't forget to like, subscribe, and turn on the notification bell to stay updated with our latest educational videos. Now, let's begin today's topic. Topics covered in this video: Electron Transport Chain, ETC. In this video, we will explore the Electron Transport Chain, ETC, and understand how cells convert the energy stored in electrons into ATP. We begin with an introduction to ETC, including its definition, location in the inner mitochondrial membrane, and importance in aerobic respiration. Next, we study the components and organization of ETC, including complexes 1 to 4, coenzyme Q, cytochrome C, and ATP synthase, complex 5. We then follow the overall electron flow from NADH and FADH₂ to oxygen, the final electron acceptor. The video explains complex 1, NADH dehydrogenase, and complex 2, succinate dehydrogenase, including their functions, electron entry points, proton pumping, and important inhibitors. We then discuss complex 3, cytochrome BC1 complex, its Q cycle, electron transfer, and proton pumping. Next, we cover complex 4, cytochrome C oxidase, oxygen reduction, water formation, and its inhibitors. We also explain ATP synthase, complex 5, and chemiosmosis, showing how the proton gradient drives ATP synthesis. Finally, we discuss ETC inhibitors and regulation, including the effects of ADP, oxygen, NADH, FADH₂, and Ca2+. The video concludes with ATP yield, the significance of ETC, and key takeaways for understanding cellular energy production. Introduction of the Electron Transport Chain. The Electron Transport Chain, or ETC, is the final major stage of aerobic cellular respiration and plays a central role in ATP production. It is located in the inner mitochondrial membrane, where a series of protein complexes work together to transfer electrons and generate energy. The electrons mainly come from NADH and FADH₂, which are produced during glycolysis, pyruvate oxidation, and the citric acid cycle. These electron carriers deliver high-energy electrons to the electron transport chain. As electrons move through the chain, their energy is used to pump hydrogen ions, or protons, from the mitochondrial matrix into the intermembrane space. This creates a proton gradient, with a higher concentration of protons in the intermembrane space than in the matrix. The stored energy in this gradient is then used by ATP synthase. As protons flow back into the matrix through ATP synthase, the enzyme uses this energy to convert ADP and inorganic phosphate into ATP. Oxygen is the final electron acceptor in the electron transport chain. It combines with electrons and protons to form water. Therefore, the ETC converts the energy stored in NADH and FADH₂ into ATP, which provides usable energy for cellular activities. The Electron Transport Chain, or ETC, is located in the inner mitochondrial membrane of eukaryotic cells. The mitochondrion consists of an outer membrane, intermembrane space, inner mitochondrial membrane, and matrix. The inner mitochondrial membrane is highly folded into structures called cristae, which increase the surface area available for energy-producing reactions. The ETC contains several major protein complexes, embedded in the inner mitochondrial membrane. These include complex 1, complex 2, complex 3, and complex 4. Two important mobile electron carriers, coenzyme Q and cytochrome C, transfer electrons between these complexes. ATP synthase, sometimes referred to as complex 5, is also present in the inner mitochondrial membrane and plays an essential role in ATP production. During electron transport, NADH and FADH₂ donate high-energy electrons to the ETC. As electrons pass through the complexes, energy is released and used to pump protons, or H+ ions, from the mitochondrial matrix into the intermembrane space. This creates a proton gradient across the inner mitochondrial membrane. The accumulated H+ ions then flow back into the matrix through ATP synthase. The energy released during this movement is used to synthesize ATP from ADP and inorganic phosphate. Thus, the organization of the inner mitochondrial membrane allows efficient electron transfer, proton pumping, and ATP production. Electron Transport Chain, ETC. This slide explains the electron donors and their entry into the electron transport chain, ETC. The ETC is located in the inner mitochondrial membrane and is the final stage of aerobic respiration. The first section shows where NADH and FADH₂ come from. Glycolysis produces 2 NADH in the cytosol. Pyruvate oxidation produces another 2 NADH, while the TCA cycle produces 6 NADH, 2 FADH₂, and 2 GTP per glucose molecule. Therefore, complete glucose oxidation produces 10 NADH and 2 FADH₂. Cytosolic NADH transfers its electrons into mitochondria through shuttle systems. The second section shows their roles. NADH donates electrons to complex 1, while electrons from FADH₂ enter through complex 2. In the third section, electrons move through complex 1 to SO-Q to complex 3 to cytochrome C to complex 4. Complexes 1, 3, and 4 pump H+ ions into the intermembrane space, creating a proton gradient. Finally, oxygen is the terminal electron acceptor at complex 4 and combines with electrons and H+ to form water. The proton gradient drives ATP synthase, producing ATP. Thus, the ETC converts electron energy into usable cellular energy in the form of ATP. Complex 1 and Complex 2 are the two main entry points for electrons into the electron transport chain. They receive electrons from the reduced electron carriers NADH and FADH₂ and transfer them toward coenzyme Q, also called ubiquinone. Complex 1 is known as NADH dehydrogenase. NADH donates 2 high-energy electrons to complex 1. These electrons pass through several electron carriers, including flavin mononucleotide, FMN, and iron sulfur centers. As electrons move through complex 1, energy is released and used to pump protons, or H+ ions, from the mitochondrial matrix into the intermembrane space. In the simplified stoichiometry commonly taught, complex 1 pumps about 4 H+ per pair of electrons. NADH is oxidized to NAD+, while coenzyme Q is reduced to ubiquinol. Complex 2 is also called succinate dehydrogenase and is directly involved in the citric acid cycle. During succinate oxidation to fumarate, FAD is reduced to FADH₂. The electrons are then transferred through iron sulfur centers to coenzyme Q, forming QH2. An important difference is that complex 2 does not pump protons across the inner mitochondrial membrane. Therefore, electrons entering through complex 2 contribute less to the proton gradient and ATP production than electrons entering through complex 1. Coenzyme Q and cytochrome C are two important mobile electron carriers of the electron transport chain. They connect the major protein complexes in the inner mitochondrial membrane and help transfer electrons efficiently from one complex to another. Coenzyme Q, also known as ubiquinone, or CoQ, is a lipid-soluble electron carrier located within the inner mitochondrial membrane. It receives electrons from both complex 1 and complex 2. After accepting electrons and protons, CoQ is reduced to ubiquinol, or QH2. It then carries these electrons through the membrane and transfers them to complex 3. Because CoQ is lipid-soluble, it can move freely within the membrane. Cytochrome C is a small, water-soluble protein located on the intermembrane space side of the inner mitochondrial membrane. It receives electrons from complex 3 and transfers them, one at a time, to complex 4. After donating its electron, cytochrome C becomes oxidized and is ready to participate in another electron transfer cycle. Therefore, CoQ transports electrons within the membrane, whereas cytochrome C transfers electrons along the membrane surface. Together, these carriers link the ETC complexes and maintain continuous electron flow toward oxygen. This electron transfer supports proton gradient formation, which ultimately drives ATP synthesis through ATP synthase. After electrons enter the electron transport chain through complex 1 or complex 2, they are transferred to coenzyme Q, also called ubiquinone. Coenzyme Q carries these electrons to complex 3, which is known as the cytochrome B.Sc complex. At complex 3, reduced coenzyme Q, or QH2, transfers its electrons through several components, including cytochromes and an iron-sulfur center. Through the Q cycle, complex 3 helps transfer electrons to cytochrome C and contributes to proton translocation. In the commonly used simplified accounting, complex 3 contributes about 4H+ to the proton gradient per pair of electrons. Reduced cytochrome C then carries electrons one at a time from complex 3 to complex 4. Complex 4 is also called cytochrome C oxidase and contains important cofactors, including copper centers and heme groups. Complex 4 is the final major electron transfer complex of the chain. Here, molecular oxygen acts as the final electron acceptor. Oxygen accepts electrons and combines with protons from the mitochondrial matrix to form water. Complex 4 also contributes approximately 2 pumped H+ per pair of electrons. Together, complexes 3 and 4 complete electron transfer toward oxygen and help establish the proton gradient across the inner mitochondrial membrane. This gradient provides the driving force for ATP synthesis through ATP synthase. Chemiosmosis and ATP synthesis. Chemiosmosis is the process by which the proton gradient generated by the electron transport chain, ETC, is used to produce ATP. It occurs across the inner mitochondrial membrane during oxidative phosphorylation. As electrons move through the ETC, complexes 1, 3, and 4 pump H+ ions from the mitochondrial matrix into the intermembrane space. This creates a high concentration of protons in the intermembrane space and a lower concentration in the matrix. The resulting electrochemical gradient is called the proton motive force, PMF. ATP synthase, complex 5, uses this stored energy to synthesize ATP. ATP synthase has two major functional regions. F0, the membrane-embedded portion that provides a channel for H+ movement, and F1, the catalytic portion facing the matrix. When H+ flows through F0 back into the matrix, the enzyme undergoes rotational and conformational changes. These changes drive the formation of ATP from ADP and inorganic phosphate. Thus, the sequence is ETC, H+ pumping, proton gradient, H+ flow through ATP synthesis, ATP synthesis. This process is called chemiosmosis and is responsible for producing most of the ATP during aerobic respiration. Oxygen ultimately accepts electrons and forms water, allowing the ETC to continue functioning. Electron Transport Chain. ETC. The Electron Transport Chain, ETC, is the final stage of aerobic cellular respiration and occurs in the inner mitochondrial membrane. Its main function is to transfer electrons from NADH and FADH₂ to oxygen, and use the released energy to generate a proton gradient for ATP production. NADH donates electrons to complex 1, while FADH₂ donates electrons to complex 2. Electrons then pass to coenzyme Q, ubiquinone, complex 3, cytochrome C, and finally complex 4. Complexes 1, 3, and 4 pump protons, H+, from the mitochondrial matrix into the intermembrane space. Complex 2 does not pump protons. This creates a high concentration of H+ in the intermembrane space. Protons then flow back into the matrix through ATP synthase, complex 5. The energy released during this movement drives the formation of ATP from ADP and inorganic phosphate. For ATP calculation, one mitochondrial NADH produces approximately 2.5 ATP, while one mitochondrial FADH₂ produces approximately 1.5 ATP. Therefore, the ETC contributes about 28 ATP per glucose. 10 mitochondrial NADH times 2.5 equals 25 ATP, and 2 FADH₂ times 1.5 equals 3 ATP. Including substrate-level phosphorylation, the complete yield is approximately 30 to 32 ATP per glucose, depending on the shuttle system. Oxygen is the final electron acceptor and is reduced to water. Electron Transport Chain, ETC, inhibitors and regulation. The Electron Transport Chain, ETC, is the final stage of aerobic cellular respiration and occurs in the inner mitochondrial membrane. Electrons from NADH enter through complex 1, while electrons from FADH₂ enter through complex 2. Electrons are transferred to coenzyme Q, CoQ, then to complex 3, cytochrome C, and finally complex 4. At complex 4, oxygen acts as the final electron acceptor and combines with hydrogen ions to form water. As electrons move through the chain, complexes 1, 3, and 4 pump protons H+ into the intermembrane space. This creates a proton gradient that drives ATP synthase, complex 5, to produce ATP. Several inhibitors block electron transport at specific sites. Rotenone and pyricidin A inhibit complex 1. Malonate and TTFA inhibit complex 2. Antimycin A inhibits complex 3. Cyanide, carbon monoxide, and azide inhibit complex 4, preventing oxygen utilization. Oligomycin inhibits ATP synthase and blocks proton flow. ETC activity is regulated by ADP availability, oxygen supply, NADH, FADH₂ levels, and calcium ions. Inhibition decreases electron flow, proton pumping, and ATP production, while increasing reduced electron carriers. Thus, proper ETC regulation is essential for efficient cellular energy production. Electron Transport Chain, ETC, ATP yield, significance, and summary. The Electron Transport Chain, ETC, is the final part of aerobic respiration and works together with oxidative phosphorylation to produce most of the ATP in cells. It occurs in the inner mitochondrial membrane. Electrons from NADH enter the chain through complex 1, while electrons from FADH₂ enter through complex 2. Both pathways transfer electrons to coenzyme Q, CoQ. Electrons then pass through complex 3, cytochrome C, and complex 4. Finally, oxygen accepts the electrons and combines with hydrogen ions to form water. As electrons move through the ETC, complexes 1, 3, and 4 pump H+ ions into the intermembrane space. This creates an electrochemical proton gradient, or proton motive force. H+ ions then flow back into the mitochondrial matrix through ATP synthase, complex 5. The energy released drives the conversion of ADP plus pi into ATP. Approximately 1 NADH produces 2.5 ATP, while 1 FADH₂ produces 1.5 ATP. Actual ATP yield can vary depending on cellular shuttle systems. The ETC is essential because it efficiently converts the energy stored in NADH and FADH₂ into ATP, linking glycolysis, pyruvate oxidation, and the TCA cycle with cellular energy production. Oxygen availability is essential for continuous electron flow.
Thank you for watching. If you enjoyed this video, please give it a like, subscribe to the channel, and click the bell icon to get notified whenever I upload a new video. Your support means a lot and helps the channel grow.